Hey, good morning and welcome to the GRCF 2023 seminar series. This morning we have Dylan Barbera. He is a global product manager for QIAGEN, and he is going to speak about microbial NGS and difficult samples, tips and tricks. So thank you. And with that, I'll turn it over to Dylan.
Sure, thanks. So like she just said, my name is Dylan Barbera, and I am a global product manager in our NGS group here at QIAGEN. Specifically, I work with all of our microbiology-based products as well as our shotgun sequencing products as well for all applications. So I'm going to just get started real quick. Right, so just before I get started, all the products that I'm talking about today are not intended for diagnostic use. They are for research use only. So just going to start off, with the tips and tricks. It's always important to remember that NGS essentially is just like an advanced PCR. So different things that will have negative effects on your PCR will also have negative effects on your NGS. So one of the more common things and more difficult sample types like stool or soil or wastewater, which we work with frequently, are PCR inhibitors. So it is always important to use an extraction method that removes your inhibitors from your actual sample as you actually do your extraction. Something else that we've recently found is that with newer PCR machines, thermal cyclers, that the ramp time of the thermal cycler itself actually will have a negative effect on your library yields. So when I say ramp time, that is the amount of time that it takes from going from one temperature to another between those. And if that actually goes too fast, you will get a lower library yield at the switches between temperatures. And a lot of vendors for thermal cyclers do actually promote how fast that their thermal cycler actually goes between these different temperatures. And we do see if it's too fast, you get a lower library yield or you get no yield at all. So I think right now we're saying that you actually do want at least 6 seconds between your temperatures to get the optimal yield for a lot of our different library preps as well. Also another thing to consider is host DNA and RNA present. A lot of people are actually surprised to hear that stool does not actually have that much host DNA, even though it is a host sample type. But a lot of things like soil or wastewater obviously do have a lot of background DNA that you have to consider. So that would really change whether you might want to do a targeted approach or a shotgun sequencing approach, or you have to do some sort of depletion, like ribosomal RNA depletion as well. And then, do you need a higher read allocation? So, another thing to highlight is more difficult sample types. If there is a higher background, we do recommend doing a higher read allocation. So on your sequencer, devoting more reads per sample. So for wastewater sample types, for example, which usually have a pretty high abundance background, they are, even with targeted solutions, there is a high background still. We typically recommend doing at least 5 times the amount of reads as you would do with a normal sample type, which is pretty much anything that isn't wastewater. So some of those things like wastewater are difficult to actually not get non-specific binding because there is a lot of non-defined black space, they call, in wastewater that we don't actually know what the microbial community even is because they're undefined bacteria and viruses. And so you do get non-specific binding with your targeted applications as well. So that's why we recommend a higher read allocation, and then you still can get high quality results if you take all of these into consideration, even from sample types like wastewater. So specifically with inhibitors, what we're kind of talking about in some sample types is like humic or fulvic acids in soil, or different polysaccharides in plants and bile. These are just things that cause an inhibitory effect on your actual PCR reaction, so that your DNA yield is much lower. And what that results in, at least in a qPCR here, is cycle times that are much later than they should be, despite the fact that you have the same copy number in those different samples, whether you've extracted them well or extracted them poorly. So how this would translate just to NGS specifically is that you would actually get a lower library yield or no library at all if you don't actually report. So a lot of times, I do see some people look at library kits and they get poor quality library yield out of there, and actually what a lot of times the problem is with the extraction itself or the quality of the sample going in. So it's always library kits are typically agnostic to, whatever the actual sample type is. So as long as you get, high quality nucleic acids out of your extraction, you should typically get a high quality library out of your library prep. So, at least for AMR, what it comes down to, your two options typically are going to be targeted or a shotgun sequencing approach. So, one benefit of whole genome sequencing is obviously that you also get the different species that are present in your sample when you're actually doing shotgun sequencing. But, the problem with AMR largely is that a lot of AMR genes are horizontal transfer, which means that you can't actually really tell what species those AMR genes came from in the first place, even if you do have the species information. So you do have to kind of correlate at best. You can also get functional profiling and all the other information you need. But, shotgun sequencing, of course, has a lot of issues with host background as well. So you're not able to see those lower abundant AMR genes, which is why you might consider doing like a targeted approach. And so for things like wastewater or your difficult sample types, a targeted approach actually could be better because there are a lot of low abundance AMR genes that you might miss in the background because of everything else that is there. You can also typically actually do a much higher level of multiplexing when you're targeting. So while you might spend more money or spend more time on your library prep upstream doing a targeted approach, you can do a lot more samples on your sequencer at once, saving a lot of money and time on the actual sequencing effort. So hybrid capture versus amplicon targeted sequencing. So in targeted sequencing, there's kind of two different approaches. One is using hybrid capture probes. The other is using, actual PCR primers and amplicon-based approaches. We found that hybrid capture actually works a lot better for viruses and bacteria as well for actually being the method to use. Part of the reason for this is that they are a lot more amenable to mutations. I don't know how familiar you are with SARS-CoV-2 sequencing, but throughout the pandemic, we regularly had to update amplicon-based panels based on how mutations were occurring for COVID throughout the entire pandemic. Because an actual primer really suffers quite quickly if there's mutations right at the primer site, especially if it's right on that 3' end, and it will just fail and you will lose an entire amplicon pretty quickly. However, hybrid capture probes do typically allow up to like a 10% mismatch in the nucleotides before they start to degrade and not really work as well. So if you are working with something where there's a very high mutation rate, like a virus or even a bacteria still, hybrid capture probes allow you to have a panel that is a bit more future-proof to analysis and having dropouts from those mutations. So, however, amplicon is faster. It is, something that you can typically do a much higher throughput for your samples. Because hybrid capture typically has, potential for overnight hybridization. So they often are 2-day workflows for your actual library prep. So if you are doing like a mass scale SARS-CoV-2 level sequencing, amplicon-based sequencing can be better because it does allow you to do many more samples at once, and through your sequencer. But if you are trying to develop something that will work for this virus for the next several years, a hybrid capture panel actually is going to be a better solution in terms of targeted genomics. So, there's several different areas that you might also introduce bias to your actual results in microbiome and other microbial samples as well. For your actual extraction, you have to verify that you have a kit that is actually breaking open all of the different bacteria that are actually present in your sample or all the different viruses that are in your sample. Extraction has changed a lot actually in the past 20 years and has gotten a lot better, where if you actually take the same samples from 20 years ago and extract them with the kits of today, you'll actually see that there's a lot more bacteria and different species of bacteria in those samples than they thought even 20 years ago, because those kits back then were not necessarily breaking open all of the different bacteria that were actually there. And if you don't break open everything, you of course are not going to get all of the results that are there. So not just QIAGEN kits, everyone's kits have gotten a lot better. So it is worthwhile to consider that as something, especially when you're looking at like old publications and things, is that all of that bacteria might not have actually been represented well in the results. Also, when you move on to actual NGS, there's a common issue called GC bias, where some library prep kits will struggle if there's a wide range of different like G's and C's in the nucleotide content, because since they do have a higher melting point than A's and T's, actually setting up your PCR reaction for a wide variety of different viral species or bacterial species, I mean, that have that different GC content can be difficult for people making like a library prep reaction. You know, you'll have bacteria that actually have like a GC content of only 30% of their genome is G's and C's. And in the same sample, you might have a bacteria that's 70% of the genome is G's and C's. And so with those different melting points, it is hard to create a reaction that actually evenly will amplify all of those different bacteria. Otherwise, some of them might appear more abundant than they actually are in your sample. So that's something else to consider when looking at kits or developing your own workflows, is how well you actually handle those different GC biases and how well they're represented. There are controls out there for actually kind of evaluating this. ATCC has a control of 20 samples that has a pretty wide range of GC bias. And it's a good way to evaluate if your reaction is actually performing properly, because they tell you how much, like what percentage the distribution is of each of those different bacteria in the control. And if your reaction's working properly, you should match pretty closely to what they got. Some kits out there really do struggle with that. And then also for analysis, it really comes down to the database that you're using, because if your bioinformatics database doesn't have the bacterial species that is in your sample, it will not report that it's there. So it might look like you have actually seen everything that's in your sample, but because maybe that bacteria wasn't categorized in that specific database you're using, it might not actually show up in your results at all. So it is important to try to use the biggest database you can, or a database that you know is good, because especially between like different publications and things, if they're using different databases, the results might not necessarily be comparable, because they actually are looking essentially not at the same thing or for the same thing. All right, so now I'm just going to move on to some solutions that we have that do work for all of these things. So QIAGEN is well known for all of our extraction kits. Obviously, that is what QIAGEN is kind of built on, but we do have downstream kits available for each of the different parts of the workflow, whether it is NGS or PCR or analysis. So first, I'm just going to touch on our X-Hive microbial panels. So we have a handful of different viral and bacterial panels that target the whole genomes of viruses. We also have a few other things in development, which I'll mention. So we have a respiratory panel, which targets 89 separate respiratory viruses and their whole genomes, and targets them deep enough to do high-level genotyping or variant analysis for each of those. We have an STI panel that targets HIV-1, hepatitis B, and 19 separate types of high-risk HPV. We have adventitious agent panel, which targets the whole genomes of 132 different viruses, but how that differs slightly from the other panels is that it is more of a detection panel. So while we do target all of those different viruses and their whole genomes, we don't have as many probes tiled into the more variable region. So when we do designs of our other panels, we actually take very many of each of the genomes of each of these viruses, and we're able to kind of compile them on top of each other and look at the areas that have the most variability. And we design extra probes that are in those regions that are more variable, so that it is, again, more future-proof to any mutations in the future. We also have our monkeypox panel, which is available in 2 different formats, where you can get it as a spike-in, which it means that you can add it essentially to any of the other panels. And then we offer like the full kit if you only want to sequence monkeypox. And then for bacteria at the moment, we have our AMR panel, which targets 2,786 unique AMR genes, which is about 6,200 plus AMR gene markers. In development, we have a hepatitis C panel, which covers the full capacity or the full diversity of hepatitis C and all of its different serotypes, and is able to actually sequence the full genome of each hepatitis C serotype. And something else that we're currently actually doing some early access testing for is a tuberculosis panel, which actually enriches and targets the whole genome of tuberculosis to allow the whole genome sequencing of TB directly from sample types like sputum or CSF. Because right now, if you want to do whole genome sequencing of TB, you have to actually do a mycobacterium culture, which is something that takes like 4 to 6 weeks if you actually get it done properly. So that will enable a targeted whole genome sequencing of a bacteria. And then next summer, we will actually have a custom version of this product available that you could actually choose what viruses you want on your panel instead of our own designs. So if these panels are too big, or if they're missing one virus that is critical to you, then you'll be able to either get like that one virus and add it to these panels, or get really any virus that you want. And so for the designs for that one, it is also doing that high level, like taking many different sequences of the same virus and compiling them together to add on to the more variable regions in terms of probe coverage. So in terms of the workflow, this is hybrid capture based. What we offer when you actually buy the X-Hive kit, which is the workflow here, what you get is the reverse transcription kit, which is compatible with total nucleic acids. So you can put in RNA, single-stranded DNA, or double-stranded DNA, and that will result in double-stranded cDNA. So really any virus type extracted can go into that kit, and that will again produce that double-stranded cDNA. We actually, in that step, include ribosomal RNA depletion built in right into the chemistry to remove human ribosomal and globin RNA as well. And then that feeds into your actual library prep kit. So we recommend using our QIAGEN FX kit for your shotgun sequencing here. However, it is compatible with any library prep kit from any vendor, not just QIAGEN's. And then you move into the actual hybrid capture. So that is an overnight hybrid capture, but it does allow for a much higher sensitivity. The entire workflow itself, from starting your RT to actually starting your hybrid capture, is about 1 full workday. And so then you start your hybrid capture and come in the next morning, and you finish up and start sequencing on that next day as well. So the only difference between any of those panels is that you switch out one tube that is here at this actual hybrid capture step. Other than that, the workflow is a unified single thing. Or if you're working with DNA only, you can of course skip the reverse transcription step. So also included in the bundle is analysis on our GeneGlobe browser-based analysis tool. So that is a more basic analysis that will give you the amount of reads for the virus detected. It will give you an alignment to best reference. But if you want a deeper level analysis, we also have pipelines built into the CLC Genomics workbench as well. Or it is compatible with any analysis tool that you want to use. So just to highlight some additional things for the AMR panel. So we didn't target any really specific drug class here. When we actually designed this, it was designed against the 4 main large open source AMR databases. So we use what we call the QMI AR database, which is QIAGEN's internal database that is kind of a conglomeration of CARD, ResFinder, NCBI, and Argonaut into one single AMR database. And we designed against all of that. So this panel does target the vast majority of the known AMR that exists, and allows you to have a much higher sensitivity for those different AMRs as well. So it allows you to see AMRs that are not ARGs that are not typically visible from just shotgun sequencing if you have a more complex sample, but it also allows you to multiplex many more samples at once onto your sequencer, saving you a lot of time on the actual sequencing runs. We typically say that you can do at least 10 times as many samples on your sequencer as compared to shotgun sequencing, but in reality, you can even do much more than that. The typical example that I give is that if you want to do deep enough sequencing on a MyCP2 with shotgun sequencing, and you want to be able to actually see your AMR genes, you can maybe run 1 or 2 samples at a time, or with this panel, at a minimum, you can do 14 samples at a time onto your MyCP2. Beyond that, since it is a targeted approach, it is kind of a faster data analysis, especially if you use the GeneGlobe tool, which is cloud-based. So this kind of difficult to see with the zoom controls over, but we actually ran this panel on a handful of different samples. I don't have it in this deck, but we also have saliva samples. And then on the left 3 is wastewater, and then the other 6 is stool samples. And you can see Wastewater, of course, is very diverse, as you might expect. The stool, and then the least diverse was saliva. So you do see like an increasing AMR, like as you go kind of downstream. What was surprising to us in the wastewater samples is we saw genes that confer resistance to the drug colistin, which is one of those more or less line of defense antibiotics, and we weren't really expecting to see it there. But in general, you do get a lot of results when you're looking at AMR and wastewater because there is just so much bacteria that is in that. So there are a handful of publications for using this AMR panel in wastewater. If you're interested in them, they're on the screen right now. If you're provided the slide deck after, you obviously can go through the time to look at them. Though actually one thing that I do find interesting actually that one of those paper was from Japan is they've actually managed to be able to sample wastewater directly at the hospital, that they're taking raw sewage at the hospital level and looking at AMR there and seeing that AMR that is specifically in an individual hospital. And then they want to be able to use that information to actually take action on removing maybe that AMR gene from the hospital itself. And the fact that they can actually sample down to the individual hospital level, I find very interesting, because when people talk about wastewater surveillance, sometimes you're at the city level wastewater, and honestly, what is the action you can take? I mean, maybe it does give information that SARS is present in your city, and then you can, start to spool up your hospitals in terms of like readiness and response for people to come in. But that is very limited in terms of the action. You're not really going to be able to close down. But if you can actually, maybe for like AMR, look at an individual hospital, maybe you actually could take actions to prevent that AMR from spreading. So just to highlight some stuff for the respiratory panel as well. Like I said, it's 89 separate viruses. I'm not going to go into each one of these, but there's SARS, influenza A, plus B, RSV-A, RSV-B, among other things. In terms of comparison to shotgun sequencing, it's kind of hard to see on this screen, I think probably to you all here, but the light blue or kind of gray is the amount of reads from shotgun sequencing in this sample, and the dark blue is the actual enrichment with this panel. And then it is a log scale here. And so we saw on average that there was a 2,400x fold enrichment compared to shotgun sequencing. For this experiment, all 14 of these viruses were actually put into a single tube at the same time and then enriched together. So this is all together that we were able to detect to this level and get this level of enrichment. And there was a background human DNA spiked into there as well. Again, and then this panel is also able to be used in wastewater. All of these different panels are used in wastewater, and we find that if you can get good results from wastewater, you can get good results from pretty much any sample type, which is why I bring it up a bunch. And it's just been kind of a continuous hot topic recently. And again, we have, publications. This is, I guess when I put this in there, a preprint, but we do, have many customers actually doing wastewater with all of this stuff. Now moving on to something that we recently released, which is our microbiome WGS seq sets. So these are bundles of both existing and new things. So at launch, we kind of have two different options. What you get when you actually purchase these seq sets is extraction, library prep, and analysis, all as one single item that is at a discount compared to buying each of them separately. So the two different options are the QIAGEN Power Fecal Pro WGS seq set or the Power Soil Pro WGS seq set for changes in the extraction kit, and then they both come with the same downstream components. So what we include is our QIAseq FX DNA library prep kit, which is our enzymatic-based shotgun sequencing library prep. We include our QIAseq normalizer kit, which is a new kit also from this year for library normalization that takes your libraries from your shotgun sequencing or any sequencing actually, and turns it into 4 nanomolar double-stranded DNA libraries immediately ready to be pulled and sequenced. And then we also include QIAseq, which are equivalent to AmpPure-XP. And then the new thing is the microbial analysis portal, which we'll get into in a second. But you get all of these for one single item, all combined together. So it's everything you need for sequencing except for literally the flow cell itself. So the FX kit is, again, an enzymatic-based shotgun sequencing kit. And what I mean by that is you don't need any sort of mechanical shearing device like Covaris. It does, take your DNA and cut it into pieces enzymatically. which is something that's very reproducible and easy to automate onto higher throughput devices. We currently have it available with 384 unique dual indexes, which of course are the different tags that you use for your samples. And then, yes, it's very compatible with automation and works very well with any species. So in terms of the general workflow, it is compatible with both PCR and PCR-free workflows. So if you are somebody who cares about PCR-free, this kit can be used for either one. It is compatible with inputs as low as 20 picograms and up to 1 microgram. So pretty much if you get any DNA from your extraction at all, you will be able to create a library with the FX kit. And then it feeds into the normalizer kit, which is an optional step if you would prefer to do, a manual normalization. Maybe you're only working with a handful of samples and it's not worthwhile to run that. But one caveat is the normalizer does require you to use the PCR workflow from the actual FX kit. So the FX kit actually won a benchmark study from the Japan Microbiome Consortium, where they took all of the different on-the-market library prep kits and compared them specifically for microbiome analysis for diverse microbial communities. And so they actually repeated this work in 11 separate labs to show how reproducible the different data is. And they chose the FX kit as the #1 kit for enzymatic library prep from that whole workflow. So pretty much any library prep kit you might have heard of was in this benchmark study, and we beat them out for a handful of reasons, to include that our kit really doesn't have any issues with GC bias, like I mentioned earlier, in terms of the different GC content that are in different bacteria. And it's very transferable across labs, and they got very similar results in all of the different labs that they ran this in, which is of course something very important for any wider studies. And they did like the fact that it was very automatable as well. So in terms of the normalizer that I mentioned, which is another new kit that just came out in the summer, obviously, if you're familiar with sequencing, you're aware that when you actually go to do your library prep, you perform the library prep self in 96 different wells if you're running 96 samples, and those do not always come out to the same concentration when you actually go to load on the sequencer. The normalizer will take all of your libraries and bring them to 4 nanomolar and have it still be double-stranded DNA libraries. Does not use any hazardous chemicals and doesn't require any specialty lab equipment at all. And it is when there's 2 different methods of the normalizer. One integrates with the FX kit, and that's a 30-minute workflow. We do have a universal method that is compatible with any kit for Illumina sequencing. That is a 1-hour workflow. But it does bring everything to 4 nanomolar, which is immediately ready to be pulled and put on the sequencer. So you don't have to do the tedious qubits or other qPCR and then manually do it. So it gets rid of that whole process there. So we've included that in the seq sets, but it is an optional step. And what that kind of results in is displayed here in these different pie charts. And the pie chart on the left is what it looks like from these different amounts of inputs if you don't actually do normalization. And then we did the normalization on the actual libraries. You can see that the slices from the pie chart here are a lot more evenly distributed, and that the CV of the different reads from the samples actually dropped significantly. And this was a pretty wide range because it was libraries that ran from 25 nanomolar to 300 nanomolar, and was able to bring them all down to similar concentrations as well. So now moving on to the analysis portal itself. The analysis portal is intended to be an incredibly easy to use and intuitive analysis for anybody to be able to do their own bioinformatics. This is really something that takes it so that anybody can do this kind of analysis on their own without the need to send it out. Now, this is included with the bundle of the kit, and this bundle is actually cheaper than buying any of those like kit boxes separately. So to you, this analysis is free. So there's no reason not to use it. It is cloud-based and browser-based, and it really is as simple as you upload your files to the analysis portal, and then you like label them with what they actually are, and you click go, and it runs the analysis, and that is it. And the analysis that it is performing is taxon ID. It does antimicrobial resistance genes as well, using that same database that I mentioned earlier, which is those 4 different databases together. And then you can also do differential abundance to compare your different samples together. And then it also, in the experiment summary in QC, will provide you with your alpha diversity with and your beta diversity as well. And all of this literally just takes 2 minutes to set up once you actually have your stuff uploaded to the portal. If you use BaseSpace, you can actually import directly from BaseSpace without having to actually upload from your computer. And you just click go and you can walk away and you come back a little bit later and your analysis is done. And all of this is already set up. And it really takes just about 2 minutes. So in terms of the taxonomic ID, it will generate charts like this for you automatically. And you can actually, from this bar here, toggle what sort of taxon level you are at. So this is selected a genus, but you can go down to species, or you can do higher things like phylums or things like that. You can export this chart directly if you want at a high quality, or if you would prefer to just generate your graphics on your own, you can download an Excel file that has all of your different reads for the different bacteria that were detected. So all of those options are available. It also does AMR. This is just an abundance chart for AMR as well. It does look very similar to the one before, but it's all of the same stuff that I just mentioned, but for AMR. If you do download the Excel file for this one, it does give a little information, more information about the given gene, like the drug that it actually confers resistance to. Some gene names obviously are very clear, like tetracycline resistance, but others are not. And then differential abundance, you can do up to 5 different comparisons graphically. You can actually have more things than 5 there, but will only actually generate this Venn diagram for up to 5. And yes, and so like I said, everything that I mentioned in this kit you purchase separately, but as this whole bundle together is the highest value. For sure, it is definitely the cheapest way to get it from us. And that is it from me. So if anybody has any questions about any part of the presentation, please feel free to ask them.
Thank you very much. I just have a question. You had mentioned the custom panels and the TV panel.
Yeah, so we're actually open for early access testing for both the Hep C and the TBE right now. Those will become available for normal commercial sale in the summer. And that is also when the custom will become available as well. So based on how things are going, the TB and Hep C might come out a little bit earlier than the custom does, but the custom will be somewhere late summer, depending on how things work out. Yeah.
I have one more.
Sure.
So you had mentioned the FX. It has been tested for automation?
Yeah, it is scripted. It is our most scripted kit for all of the different vendors. So pretty much any vendor that sells a liquid handler, we have scripting for, and if it doesn't, typically we're willing to help you work with scripting vendors for it. So what do you have a specific?
I was thinking, yeah.
Yeah, TCAN. Well, I guess there's different robots, right? Do you have a Dream Prep or yeah? Yeah, we have a couple different TCAN scripts. Okay. So it depends on which one, because those robots are so different from each other that.
Yeah, it's great to hear that. Yeah.
From our virtual audience, please raise your hand and I'll be happy to allow you to speak. Okay, and there are stuff in those. So thank you very much, Dylan. We appreciate you coming today and sharing this information with us. We look forward to